And we will move then forward to the next speaker and the last speaker for today, which is Carolina Monzó, from the Institute of Integrative Systems Biology at the Spanish National Research Council. And she will talk to us about dietary restriction that mitigates the age-associated decline in mouse B-cell receptor repertoire diversity.
Thank you. So, hi, everyone. I'm Carolina Monzó. I am currently a postdoc at the Spanish National Research Council, but this work that I will be presenting today was done during my PhD at the Max Planck Institute for Biology of Aging. So, okay. You will probably know this, but aging is a progressive loss of homeostasis and a failure of body functions that leads to development of associated comorbidities. And these include many things. Among them are dementia, cardiovascular diseases, and cancer. In 2013, Carlos Lopez and other researchers proposed the hallmarks of aging. These were 12 ideas that they had, and then after 10 years of research in the field, they have revised them again. So these 12 hallmarks of aging, I was especially interested in this pink part right here. This includes the microbiome, the immune system, stem cell regeneration, and other topics. And it was interesting for me because we know that during aging, the adaptive immune system undergoes a profound dysregulation, and this results in impaired responses to pathogens and also vaccinations. So with this interplay of immune system and microbiome in mind, I was interested in understanding how the B-cell receptor repertoire, or the BCR, changes with age, as these receptors are indispensable for recognizing and binding to antigens. So what we know so far in age is that in old age, there is a loss of repertoire diversity. There is increase in clonal expansions and also impaired primary and clonal B-cell selections, as well as affinity maturation. And you know this, but when a B-cell is exposed to antigen, they need to generate a response to antigen and also create memory. And this happens by clonally expanding the B-cell with the membrane-bound antibody of interest. And as you can see in this, hopefully you can see, yes, the cartoon, this is what we understand is going on with age. If different naive B cells are these different colored dots, and this different color part down here corresponds to the clonally expanded portion of the B cell receptor repertoire between individuals. In old age, the naive B cells are displaced by antigen-experienced B cells that come from these clonal expansions. So there are less naive B cells, less B-cell receptor diversity, less capacity to recognize different antigens. And this impaired immune function increases the aging phenotype as well. So we have more poor health and high frailty. A great strategy to improve health at old age is by improving the aging process itself. So we thought it was important to discover whether anti-aging interventions ameliorate or reverse the loss of the B-cell receptor repertoire diversity and increase in clonal expansions that have been reported previously. So dietary restriction, which is reduced nutrient intake without malnutrition, is the most effective anti-aging intervention to extend lifespan and health span in multiple animal species. As you can see in this study in mice, it can extend lifespan by up to 40%, and it can ameliorate morbidity and mortality in old age. Previous studies have shown that dietary restriction maintains production of naive T cells and T cell receptor repertoire diversity in primates, but we don't know how dietary restriction affects the B cell receptor repertoire diversity. Also, late onset dietary restriction can improve health, and this avoids the need for long-term DR feeding, which would be more translatable to humans, because obviously I want to start dietary restriction as late as possible and still have this very good phenotype. And for example, in this study, onset of dietary restriction at 15 months of age, which is late middle age in mice, improves regeneration of aged hematopoietic stem cells. So we are also interested in exploring the effects of dietary restriction starting both early and late in life on the diversity and clonality of the B-cell receptor repertoire. To understand how this BCR repertoire composition is influenced by aging and dietary restriction, we isolated RNA from the spleen and the ileum of female mice because we wanted to capture the systemic and also the gut-specific profiles of the diversity of the BCR. We used 5 mice per treatment at 5, 20, and 24 months of age. So we would also obtain a temporal profile. And the treatments that we used were wild-type ad libitum mice, so they could eat whatever they wanted. Mice with dietary restrictions since weaning at 3 months of age throughout their whole lives. And then mice switched from ad libitum to dietary restriction at 16 months of age to see these effects of late onset DR. We limited our analysis to the BCR heavy chains since they were reported to be sufficient to identify clonal relationships with high confidence. But I will look into the light chains after Ken's talk. But this was enough for us to study diversity and clonality. We then used this extracted RNA for sequencing the BCR receptor repertoire. We retrotranscribed to cDNA using a template switch that added a UMI, and also building on top of isotype-specific primers that were binding to the constant chain. Next, we ligated the adapters. So this allowed us for unstranded binding. So when we performed asymmetric sequencing, 400 times 100 base pairs on an Illumina NovaSeq, we could see reads that we would be able to later on collapse and have this 600 base pair fragment fully sequenced from side to side. So from now on, we fully worked with the Immcantation tools. We used Presto to perform all the data preprocessing. This includes quality filtering, the multiplexing, because we had also internal barcodes. We generated consensus read generation from collapsing the reads that had common UMIs. And then we assembled the read pairs by merging the reads into a contiguous sequence, which is basically what I was showing here. And then also, since we had the information of the primer that was bound to the constant chain of each of the sequences, we could also annotate the isotypes of the heavy chain. Then using IG-BLAST, ChangeO, and the reference mouse, B-cell receptor repertoire from IMGT, we annotated the B, D, and J genes. And finally, since accurate identification of clonal relationships is crucial to study the repertoire, we needed to find the appropriate distance threshold for clonotype assignment. So we would be seeing what is clonally related and clonally not related. So they belong to independent clones. And these distance thresholds, we calculated them either using hierarchical clustering with Shazam, when the distributions were bimodal and very clearly separated. And in the cases that the distributions were not so clear, we used Scopper, which is built on top of spectral cluster. And then using these distance thresholds, we used ChangeO to define the clonotypes, and we organized our process data into a data frame for analysis. Also for analyzing our data, we used Immcantation tools. So here we used Alakazam to measure the diversity of the BCR receptor repertoire. And this diversity is defined by the number of clones, the relative frequency of the clones, and the subdivisions of the clonal population. So basically, mostly how many expanded clones there are and how much of the total repertoire they represent. To study this diversity within individual mice, we calculated Hill diversity spectra, and then we extracted the diversity order values that corresponded to Shannon diversity and Simpson diversity. So Shannon is a measure of clonal richness and population structure that is mostly affected by rare clones. And then Simpson is another measure of diversity, but it is mostly affected by the larger clones. As you can see, we saw an age-related decline in both diversity indices in ad libitum mice. These are the blue lines. And although there was a decline in the dietary restricted mice as well, very clear, it was significantly less pronounced. And this to us looked like the DR was maintaining a more uniform distribution of large clones and small clones. And importantly for us, humans who want to delay doing dietary restriction as much as possible, the late onset dietary restriction group recapitulated the lifelong DR's Shannon diversity. And although not the symptom, it was still quite encouraging for us that we had this partial convergence towards the R-like levels. With all of this in mind, we wanted to also determine whether the age-dependent decrease within individual BCR diversity went hand in hand with a B cell population that would be skewed towards clonally expanded B cells. So we used Alakazam to calculate the clonal expansions as the percentage of the B cell receptor repertoire that was taken up by the 20 most common clones. We saw that clonal expansions increase progressively with age in a Dietary mice, of course, and also in DR, but again, to a significantly lesser extent. This was already confirming this within individual diversity clonal expansion relationship. And again, very encouraging because we saw that DR onset at 16 months, so the later onset group, reduced the clonal expansions in the repertoire to the same extent as the DR, as you can see clearly here. We next use the RDI package to evaluate inter-individual dissimilarities. So these are differences in clonal composition of B cells between individuals, and they must be accentuated by the proliferation of different clones in different individuals. Basically, going back to this cartoon from the introduction, expanded clones overrepresent a set of sequences that will be different by default between individuals. So if each individual has a different expansion represented here by the different colors, there is a bigger difference because only very few B cells have the chance to be similar between each other. And as we expected from our findings in diversity and clonal expansions, we saw that inter-individual dissimilarity was progressively increasing with age in ad libitum and also in dietary restriction animals to a lesser extent again. And this was telling us that DR was mitigating this inter-individual dissimilarity effect. And also the late onset dietary restriction mice were recapitulating the DR phenotype. This highlights the attenuated age-related increase in inter-individual similarity, this relationship that we are observing. Then we continued our research with of the BCR by studying somatic hypermutation, which is the mechanism for affinity maturation of the BCR in response to antigen exposure. We calculated the mutation frequency using Shazam. This is from the Immcantation, of course, and consistent with previous research, we did not find any differences in synonymous or non-synonymous somatic hypermutation frequency with age or with diet. Then we calculated CDR3 length, which has been previously associated in a causal manner with autoimmune disorders and also with old age. And we did not find any age or diet effects on the CDR3 length. And then to query the repertoires on whether their class composition changed affected by age or diet, we divided the clones into 3 groups. Those with no IgM or IgD where they had B cell receptors that had already fully switched, and we thought of them as post-antigenic. Those that still had IgM and IgD but had a somatic remutation rate below 1%, so non-mutated, were considered naive. And then those that had IgD and IgM and were also heavily affected by somatic mutation were antigen exposed for us. And also surprisingly, in this case, when we were comparing between age and diet, basically vertically in this plot, we did not find any relevant differences between mice of different ages or diets. Then, since we had performed all of this analysis on spleen samples, we ran the same analysis on the mice guts. The reason for this is that dietary restriction modulates the composition of the gut microbiome in mice and humans. In this case, we are working with mice. And since mucosal B cells are in contact with the gut microbiome, we expected to see effects on the ileum, which is the region of the gut that contains the most amount of B cells. They are found in like Peyer patches, they are called. very surprising, but we did not find any changes in diversity, in clone expansions, interindividual dissimilarity, CDL3 length, or even class composition. However, we did find some significant differences between attributum and DR in somatic permutation, the non-synonymous hypermutation. We saw that DR mice had significantly higher non-synonymous mutations introduced by somatic permutation. And this was interesting because what we were understanding from it was that maybe the homeostasis with the microbiome was maintained by diversifying the existing memory B cells instead of generating new B cell clones. So in the ileum, DR was associated with intact B cell selection and elevated somatic mutation in old age. But unfortunately, this effect was not recapitulated by the late onset dietary restriction mice. Too bad. And since we only observed mild effects of the dietary restriction diet on the repertoire in the ileum, we were interested in uncovering also whether the intestinal microbiome composition and the repertoire, the B cell receptor repertoire of the ileum, were uncoupled. We thought this might be true since plenty of previous work has reported significant changes with age on the microbiome. And this happens in terms of diversity, loss of beneficial bacteria, accumulation of pathogens or commensal bacteria. And it has already been shown that dietary restriction positively affects the intestinal microbiome by maintaining high abundance of bacteria that are positively associated with health. So to directly address this question, we performed 16S sequencing on the hypervariable region 4 of the cecal pellets from exactly the same mice that we were studying the B cell receptor repertoire. What we observed were significantly higher interindividual dissimilarity in attributum mice compared to both dietary restriction and the late onset DR group. And this was confirming our suspicions that the cecal microbiome is more susceptible to change caused by the diet than the BCR repertoire, but these two things are uncoupled. Yes. And then since we had calculated so many B cell receptor repertoire metrics, we wanted to try and understand how they are reflected in the host health. And to do this, we developed a sort of macromorbidity index. We adapted it from other sources, but made it to encompass the collected macropathology of these mice. We had been, every time that we dissected a mouse, we were writing down all the phenotypes that were observed and those that were not found. And what we saw was that our mice had tumors in connective tissue, liver, and lungs. But there were also many non-neoplastic pathologies, such as bad habitus, kyphosis, enlarged spleen, discoloration of white adipose tissue, and other things like this. So what we did was we graded neoplastic pathologies where 0 was the absence of tumors, 1 meant one organ is affected by a tumor, and 2 happened when 2 or more organs were affected by tumors. So this would be like metastatic cancer. And then for non-neoplastic pathologies, we simply assigned a degree of 1 to each pathological finding at dissection because we could not really tell how much of an impact each one of them had. And with all of this information, we calculated this sort of macromorbidity index, which was the sum of the non-neoplastic pathologies and the tumor degree that we had assigned. Then we performed linear regressions and Spearman correlations between each B cell receptor metric that we had studied using the Immcantation software throughout this project. And we regressed this against the calculated macromorbidity score. We found that the spleen's clonal abundance, individual dissimilarity, and CDR3 length were positively correlated with macromorbidity, meaning that the values of these metrics were high when the mice were more sick, while Shannon and Simpson diversity, they were negatively correlated with macromorbidity. So higher within individual diversity is correlated with a healthier state in our mice. And as you remember, we found lower clonal expansions and repertoire dissimilarity in DR mice paired with increased Shannon and Simpson diversity. And this suggests that DR delays the systemic functional decline of the B cell receptor repertoire and is associated with a younger or healthier phenotype. And similarly, we had found decreased clonal expansion and repertoire dissimilarity index in the late onset dietary restriction group, also with increased channel diversity. And this was also suggesting that initiating dietary restriction later in life could recapitulate the profile of the dietary restriction repertoire and be correlated with lower macromorbidity. So just to summarize what I have been telling you, we saw that dietary restriction delays the age-associated changes in the splenic BCR receptor repertoire. Also, the splenic BCRs respond to a late start of DR and recapitulate most characteristics of lifelong DR repertoires. We also saw that the BCR receptor repertoire of the ileum is only mildly affected by age or diet. And interestingly, it is uncoupled from the microbiome diversity, although they are in direct contact. Finally, we saw that DR and late onset DR mice have repertoire metrics correlating with healthier macromorbidity scores. Basically, our observations in this project highlight that even when started later in life, a short-term dietary restriction treatment can have beneficial effects on the adaptive immune system in mammals. And just to wrap up, I would like to thank Professor Dame, Linda Partridge, Sebastian and Sonia from her lab, and our collaborators, Dario and Andreas. But most importantly, the Kleinstein lab and the collaborators for developing the Immcantation framework, since this project is fully based on top of what they have built. And if you're interested in learning also about the observations in all the isotypes, which we also calculated, you can find them in this paper down here. And thank you, and I will be happy to take your questions.
Thank you very much, Carolina, for this very interesting talk. Maybe I can start with a question. Which type of dietary restrictions have you considered? And is it just based on calories, or also the type of foods also plays a role there?
Yeah, so our lab, Linda Partridge, she has been working on dietary restriction for many years. And the way that we do dietary restriction is simply so we don't change the macronutrients. It's the same pellet. We just weigh how much the ad libitum mice are eating, and we reduce this by 40%. And this is what we give to the dietary restricted mice. And so basically they eat once a day, unfortunately. And yeah.
Nice. Okay, so yeah, we should take example for our late life, then if it's also is applicable to humans. Would be interesting to know, of course. There's further questions in the chat, which we encourage you to also answer. But we will, I think, now proceed then to wrap up the meeting. Thanks a lot again for your presentation.